Hello, bio students. This is just a quick video I made to discuss the differences between mitosis and meiosis and to help you with the homework for tomorrow. So we learned last unit that in our cells, like maybe our skin cells, our liver cells, our blood cells, our neurons in our brain, that we have a location in the cell called the nucleus, and in there are chromosomes. And chromosomes are big, long segments of DNA that we now know have 4 different nucleotides, A's, D's, G's, and C's. So A, T, G, and C are the 4 nucleotides that in any order can create a gene, which codes for a protein, which we then see as traits. So something to note is that in any organism, there's always 2 copies of each chromosome. In humans, we have 46 chromosomes, so that means we have 23 pairs. And each chromosome pair is going to have a mom and a dad gene. And we learned that during the cell cycle that we duplicate chromosomes. So this chromosome right here has 2 copies and 2 copies that are divided into 2 identical daughter cells. And these cells are called somatic cells because they are cells that don't create eggs or sperm. So every cell in your body that isn't a sperm or an egg cell are somatic cells. And they're called diploid cells because they usually have 2 copies of each chromosome. A maternal and a paternal chromosome.
I have a wonderful surprise for you.
Recording a video for kids on meiosis, so can you wait a minute?
Meiosis!
Meiosis! So this is the AP Bio students. So let's talk about testes and eggs for a minute.
What?
Notice,
We're out.
right here we have 4 chromosomes, 2 pairs of chromosomes that are homologous. So this maternal and paternal chromosome are homologous, and this are homologous. And we call them homologous chromosomes because they're the same chromosome. One is for mom and one is for dad. They code for the same genes, but they might code for different traits. So if this was the gene for hair color, this might code for red hair and blonde hair, which means that you might express red or blonde or somewhere in between. So this is my diploid cell that is going to eventually become haploid gametes, either sperm or egg. Now during meiosis 1, we first duplicate each chromosome. So I still have 4 chromosomes, 1, 2, 3, and 4. But I have 8 chromatids because I have 2 copies, 1, 2, 1, 2, 1, 2, 1, 2 of each chromosome. The other way I can think about this is I have 2 pairs of homologous chromosomes lined up on the metaphase plate. So right here is a pair of chromosomes that are homologous. They code for the same gene, but they might code for different traits. And another homologous pair here. 2 homologous chromosome pairs, 4 chromosomes, 2 that are paternal, 2 that are maternal. We separate them, and then I have 2 cells that are haploid because now they have 1/2 of all of the chromosomes because we went from 2 each to 1 each. So here I have 2 chromosomes, but 2 copies, and 2 chromosomes and 2 copies. One of these is mostly maternal, and one of these is mostly paternal. During meiosis 2, I separate sister chromatids, which are duplicated copies of the same chromosomes. So this chromatid will go here, and this chromosome will go here, and same for here, and here, and here. My end result is that I have 4 haploid, meaning half of the information, cells with 1 chromosome and 1 copy each. But that's not all. There are 2 ways that we can make genetic diversity occur at a really large scale when it comes to meiosis. The first is independent assortment, which is basically that each homologous chromosome pair can line up independently of each other during meiosis 1. So notice on this homologous pair, I have a gold and red, and then a gold and a red. They're on different sides. The number of possibilities is always going to be 2 to the power of the number of pairs of chromosomes. We have 23 in humans, so this results in 8 million different combinations of red and gold, or maternal and paternal on each other side. The other is called synapse or crossing over. This is where random but specific identical parts of chromosomes, swap between mom and dad chromosomes during meiosis 1. So you'll notice this gold region from the mom chromosome swapped with this red region of the dad chromosome. This results in gametes that aren't just mom or dad, but mom and dad combinations, which helps us create much more genetic diversity than just crossing over, or I should say independent assortment. I hope this video has been helpful. We will talk more about this tomorrow, and have a great Wednesday. See you soon.